You don't have those yet, so I'm going to talk a little bit about a lot of the work and the main work that I've been doing in the lab, and maybe this will give some inspiration for how you can think about and approach the questions and the problems that you've been asked. So mainly, I've thought about nanopore direct RNA sequencing, which is, as you've heard from many speakers, is a novel technology that's able to sequence RNA directly. And to focus specifically on this, what are you trying to really capture in a nanopore direct RNA sequencing experiment or in an RNA sequencing experiment today? Well, ideally, any technology that you would use would allow you to capture the full length of an RNA molecule, would allow you to capture the full poly A tail or other end modifications of a specific molecule, and would ultimately allow you to capture the full sequence. And not just the four canonical nucleotides, A, C, G, and U in the case of RNA, but also the modified nucleotides. There exist over 200 epitranscriptomic modifications. The most common are shown on the screen, and they exist in different proportions in a wide variety of RNA species found throughout the cellular transcriptome. And ultimately, those epitranscriptomic modifications have been linked through various studies to a plethora of different mechanisms, which are thereby implicated in multiple disease states, conditions, and pathologies. So there are many technologies to look at the epitranscriptome on a single molecule level. So Chris has previously given a lecture, talked about some of the earlier technologies that this was where we looked at the epitranscript, where his group looked at the epitranscriptome in particular, but everyone was really trying to get to this idea of single molecule. Can you see on the molecule itself the relationships between m6A and other modifications up or downstream from it? And so, there's all of these chemical strategies that Chris mentioned, which, are able to use some kind of chemical treatment to either protect or deprotect selectively the modification sites that have the modification of interest and then allow you to read it out within a next generation sequencing experiment, such as even with Illumina short read. Nanopore direct RNA sequencing, though, is the only technology, if we think back to our original goal, that might be able to tell us the full length as its long read, might be able to capture a poly A tail, and give us the full sequence. What all of these other technologies lack is the ability to profile multiple modifications at the same time. Once you start doing too many chemical treatments, right, the risk of interference or otherwise grows stronger. And what's worse than having data? Bad data, right? We want good data. So nanopore direct RNA sequencing is at the moment the best choice to look at this question and to try and do RNA sequencing today. So it begs the question, how is the technology actually doing? We have the three criteria right in front of us: full length, full poly A tail, full sequence. And let's get started. So what's the promise of full length that we can capture these tails? Well, something that Nanopore does really well is capturing the 3' ends of molecules. And this has to do with the fact that the assay is a poly A pull down assay. So you can imagine basically a poly DT primer capturing RNA species, being ligated with a motor adapter, pulling it into the pore, and then pulling it from the 3' end to the 5' end. So you can get a very precise readout of 3' ends that are captured. But as you can see, if you look over to the right, there's a more variable 5' end. This is because nanopore direct RNA sequencing exhibits 5' end truncation. So what you're looking at right here is an animated GIF that repeats on loop, which demonstrates the RNA calibration strand. The RNA calibration strand is a 1,315 base pair yeast enolase, it's from the yeast enolase 2 protein coding region. It's a piece of RNA that is synthesized and gets spiked into every nanopore direct RNA sequencing run, unless you actively choose to exclude it. It's a default part of the protocol and allows you to do some quality control on your signals, on your modification calling, etc. And ultimately, right, so on the right is the 3' end, on the left is the 5' end. You see that as you go, there's sort of this random fall off of the reads. And there's a lot of coverage on the 3' end and not as much coverage on the 5' end. And that's because as the RNA molecule gets pulled through the pore, 3' to 5' end, there's a random chance that that molecule is going to fall off and ultimately lead to a truncated read output. So this is sort of a feature of the data, something to keep in mind when you're looking at the relative proportions of reads on either the 5' or the 3' end. It's going to be heavily affected by this technical artifact. And the modern chemistries have made this somewhat better, but no matter what data you look into, this is going to be a feature of it. And if you were curious, I know you were probably all counting. If you were curious how many reads were on this slide, that's about 1.3 million. IGV, though, only subsets it and randomly samples, I believe it is approximately 50,000 to show on screen. So what are the solutions to this 5' end truncation? Well, one is to look at known annotations, known 5' annotations, and try and subset for reads that match those known annotations. This strategy will generally, right, if you have a shorter transcript, so if you have something that's 2 kb, you're more likely to capture then reads from that particular region than you would be for a 50 kb transcript, which might have a lot of coverage, but will have very few reads that will have extended all the way to 50 kb. So that is one strategy. And then the second strategy would be to try and predict, right? We said we wanted a technology that would allow us to predict epitranscriptomic signals. And the cap of RNAs is itself an epitranscriptomic signal with a very defined pattern, at least for eukaryotic mammalian caps, for those different modifications. So if you were able to predict the M7G signal at the cap of that RNA, from the signal, you would be able to also have a solution for finding reads that fully transversed through the pore. And if you're interested in thinking about this question more, that is also the assigned project I2. Great. So that's how we're doing on full length. Very good at the 3' end with some deficiencies at the 5' end. What about poly A tails? Well, if you look at this graph again, and you look at all of those turquoise blobs that I've circled, those all represent poly A tails because the assay is poly DT primed. You first capture all of the signal from the poly A tail and then incorporate it into the assay. So the way that Nanopore works is based on Kamer models. So within an individual pore, there might be 9 base pairs or up to 20 base pairs, as some of our previous speakers have alluded to. And these are all associated with a specific current signal. That current signal represents basically the voltage difference as that characteristic Kamer traverses through the pore. So the AAA has its own characteristic Kamer signal that you can see on the screen, and you can basically utilize that in order to measure and examine the poly A tail. When you look at an actual RNA signal, as shown here, because the poly A has no variation within the actual Kamer that exists within the pore, you can basically see that there's a very flat, there's almost no change in the mean current signal, and then it stays constant over a period of time. And that dwell time for the poly A tail in the pore allows you to estimate the poly A tail the poly A tail length. And this is
And
something that is a part of.
so there's
Go
no
ahead.
y-axis, but it's picoamps, I think, the current that's there to just to highlight how it's very small amperage changes.
Yes. So yeah, so that's measured in picoamps and then converted. Well, if you remember back to the Slow 5 tools lecture, we talked a little bit there about the conversion between this picoamp signal and what is actually reported by Nanopore and the hardware reasons that underlie that conversion. So that's the poly A tail, which actually, right, seems like a pretty decent progress. We can capture full-length poly A tails and measure them. Now the full sequence, probably the trickiest problem because this is representing the canonical nucleotides and the modified nucleotides. So the first thing is that nanopore direct RNA sequencing has gotten much more accurate since the original chemistries and now allows for SNP profiling and calling. And so this actually relates to project I-15. And you can basically call these single nucleotide variations within the sequence directly from the RNA data as long as there's coverage in that region. So you're not basically doing a germline or a genome variation search, but you can, if there's RNA or expression at a certain point, capture variations at those points and then compare between samples, between species, between people, whatever your comparison groups are for whatever question is that you want to ask. And then ultimately, what we want to do is try and relate some of the SNP calling and genomic information to changes in the epitranscriptome and the like. So high throughput epitranscriptomics relates to this concept beyond SNPs, beyond the four canonical nucleotides, of being able to include and call in high throughput fashion epitranscriptomic modifications. The question is, right, how many controls do we really need for a single experiment to generate data that we can analyze and that we can compare and that we can understand? So in the sense of an RNA-seq experiment, every time you do a new RNA-seq experiment, you run a very standardized protocol and you get expression, gene counts, outputs, and the like that you can use that are readily interpretable. Whereas, in other contexts, right? What if you needed a control, right, for all of the technical aspects of the way that you made the RNA sequencing preparation library? It would be very hard to scale and apply globally to larger questions and to many different contexts and studies. So we started envisioning, right, the nanopore direct RNA sequencing as sort of following a similar trajectory and outputting not just gene counts, not just SNPs, but all of the various epitranscriptomic modifications and associations. And this is something that we sort of piloted with the astronaut study paper with software that since has been far surpassed by other research, open source research tools that have been developed by research teams and the epitranscriptomic models that have been released by Nanopore. So we, in that paper, used M6A net. And there's a lot to say about this chart, but I'll just say if you look at M6A net, it might not look as great as you'd want in this sort of benchmarking study. So that they looked at glory data. So glory data is one of the chemical modification techniques to get single molecule site level, ultimately aggregated as site level, so a single adenosine nucleotide and a single position within the genome data and knockout, so metal 3 knockout, removing the writer from M6A. If you have no writer, you're not going to get a lot of M6A. And what you see is there's a ton of false positives, and the true positives also have are lacking a bit. So there are a lot of these open source research tools and frameworks that have been released, but in addition to these sort of more unofficial third-party research groups, more recently Nanopore has released their own models. So the first M6A Dratch model, Dratch meaning that it predicts M6A within specific 5 nucleotide motifs was released at the end of 2023. 2024 was very exciting because London Calling saw the release of the m6A all context model. That means it tries to predict m6A in every single model, and the pseudouridine model, which is another modification, also all context, looks at every single uridine and predicts on it. And then on September 17th, 2024, at the Boston Community Meeting, Nanopore released the 4 all-context RNA models, in addition to an updated m6A Dratch model. And so, the time, the place for multi-modification RNA base calling is here. And we're very excited. We have a lot of Nanopore direct-RNA sequencing data in the lab. If you noticed from the projects, that doesn't even begin to cover the range and variability of what we have. And it's very exciting to just jump in and look at all the data and look at all the different contexts and differences. But before you can, I guess, get to biology or novel biology, you always have to ask the question, what kind of technical benchmarking do I have to do first to see whether this works for the kinds of questions that I want to ask? And I can show you here that, right, Nanopore has these specific claimed accuracies for, how often they miscall A's or m6A's, the false negative, the false positive rate. The way that they do these calculations are on a single molecule level. So molecule by molecule, and they know, right, on that specific molecule, on that specific place, in that specific point, they have synthesized, right, an m6A. And that m6A will be present in 100% of the molecules at that specific site. And they use that for, A's as well to basically have their negative and their positive controls. And they generate these per molecule accuracies for the different models. So for m6A, I believe that this first model here is the m6A Dratch model, 95.62% accuracy. The pseudouridine model, 96% accuracy. So m6A inosine is a combined model because m6A and inosine both would be predictions on adenosine. So the modified model basically works by saying, okay, for every adenosine, is it a canonical adenosine, an inosine, or an m6A? And then M5C, which is has a little lower of an accuracy rate, 85.37%, a little harder to piece out. And you can see, right, what's sort of next on their modification hit list and the kinds of things that we're expecting going into 2025 and 2026. So these are the claimed per accuracy rates, but that doesn't mean that we're done with the technical benchmarking and something that we need to sort of explore and figure out ourselves. So the next, however long I have to still present, we'll be going through some of these sort of side quests and understanding how we can verify these claimed accuracies and how we can understand their per molecule accuracies, what they mean for our data and for our examples. So I had mentioned glory, and just to mention that glory is sort of important because we're looking at m6A. M6A is the most common modification in the eukaryotic genome. It accounts for approximately between 0.1 and 0.4% of all adenosines. And so having ground truth data produced with glory to verify our m6A predictions and our looking at m6A is important. So we used MESC, publicly available MESC glory data for a lot of these quantifications. And then we used mass spec, as you can see, to verify the loss of m6A specifically in these green samples. So this is in collaboration with Sammy Jeffrey's lab. So if you look at these green samples, the wild type MESC has a lot of m6A, and this one specific metal 3 knockout has resulted in a verifiable loss of m6A. So these two samples were sequenced on RNA-004 on a low coverage, in a low coverage manner. So that means we have about 100, 200,000 reads per sample. We later resequenced them on a higher sample. And again, we want to do benchmarking. So this is the first iteration of benchmarking. The next few slides will be probably from the last summer. So as you can see, last summer we only had the m6A all context and the pseudouridine all context. But I think the story, even though it's, these models are a little bit older, the story is still very useful for understanding how we sort of arrive at our conclusions and how we learn things. So this is basically the wild type versus metal 3 knockout comparison using this Dorado base calling 0.7.0. And this is only, so each dot is a specific site, and only sites that had at least 10 reads were included for the fraction modified. And if you were thinking that this doesn't look good, if you had that little inclination, you were right. If there's a lot of, sites in the knockout, that doesn't look very promising. But what was, and if you look at the histogram, it's also pretty scary that most sites, kind of look the same here. This is sort of our initial inclination. But the good news was there are these like true positive sites that you can identify that exist in the wild type and don't exist in the knockout. There's not a similar population in the knockout for us to compare. So the question is, what can we learn by looking at all of these different buckets, right? If we look at the bucket of sites that have more than 80% stoichiometry in the wild type but don't in the knockout, if we look at the sites that are the same, if we look at the sites that are below 80%, if we look at the knockout 80%, what can we sort of learn by sort of isolating these groups, trying to go back to first principles of the technology, which is these signals and these currents, and understand what's going on. And I already sort of showed this slide, but it's important to show again, which at the end of the day, this is a bunch of squiggles and a bunch of currents. And if we can try and parse that out with our biological understanding, crucial understanding, it just sort of becomes a matter of statistics. So one sort of sanity check was I've kind of mentioned this Dratch motif. This is the more formal definition of the Dratch motif from Sammy and Chris's paper on it. But sort of this motif where m6A is commonly deposited. And it kind of makes sense that if you have a real true positive, it'll probably be in a Dratch motif. And if you don't have a true positive, probably won't be. So luckily for us, that was the case. About 50% of the sites that popped up in the true positive bucket, those were enriched with Dratch motifs and the other things weren't. So when you think of these signals, this is going a little bit deeper into the F5C content, which is the tool that I actually use to do this analysis. But aligning, right, a signal to a sequence is not a necessarily easy computational problem. But Hasindu sort of implemented this approach for, being able to take again, utilizing this Kamer model, knowing what the mean current pores are, and being able to remap the original signal to each individual Kamer. So in this context, each Kamer gets 3 specific parameters associated with it, in addition to the associated signal, right? So for this particular Kamer, G, G, T, A, T, T, for this specific Kamer, you can extract, right, each individual point that was measured, whether it's 50 points in the signal, 100 points in the signal. But you can also summarize this data as looking at the event mean, right, which relates to the picoamp current, which was predicted by the Kamer model, the event length, so how long it's predicted for this specific Kamer to have stayed within the pore, and the deviation in the signal that's associated with the pore. So all of these summarizations can be used to try and explore individual Kamer signal currents on that level. So there's a couple sort of questions as to, why aligned reads may fail when you undergo this sort of signal to sequence conversion. So if you have, right, a signal and it did get base called into a sequence, will it always be the case that you can realign the original signal to that sequence? And the answer is, in theory, yes, but practically that doesn't always work out. And this was a little bit of exploration as to why. And this is sort of ultimately a lesson in like data transferring, management, data processing, just because if there's a particular problem that might be too difficult, maybe that isn't a function that's supported, even if, right, the claim is that we can align every single signal, every signal to every sequence. And so what's sort of happening here, this discrepancy, is that for some of the reads, the difference between the pore model, what the pore model predicted, and the current signal actually was, and the ultimate like K-mer, like the sequence prediction from that signal, that was too great. So you lost certain reads as a result. The other reason for this is that you could have, so in addition to this miscalibration phenomena, you could also have the split read phenomena. So when the Dorado base caller takes in a signal, it will actually search that signal for the Oxford nanopore specific adapter sequences that are in front of the poly A tails. Typically, right, these get chopped off, right, and you would only expect there to be one adapter basically hooked on to one poly A tail and then an RNA molecule. But what can sometimes happen, and it's rare, is that you get an adapter, poly A, an RNA molecule, and then another adapter that hooks onto the front of the RNA, poly A, RNA molecule. So it looks for the specific RNA adapter sequence and is able to split reads based on the presence of multiple RNA adapter sequences within what it recorded as the same signal. It's unclear whether there was a physical, right, connection between the front of one RNA molecule and the RNA adapter of another one, or whether this has more to do with the fact that you very quickly, right, you traverse one RNA molecule through, it goes through, and then right away, right, a new RNA molecule and a new adapter and a new pore starts to flow through. And it doesn't have enough time to separate, or reset the two signals, and it records them as one. So that sort of split read phenomena is something that Nanopore, the base caller does internally, but not something that F5C supports because the split read ultimately get different read IDs, and whatever the other split ID is, it's hard to remap that, right, back to its original signal. So originally, Dorado didn't even support, they've added now, since 2024, a new flag that allows you to look at the parent read IDs in the signal and reconnect them up. But still, even with that new addition into the base calling algorithm, you can't remap with F5C. So if you look at, okay, how many like reads are like miscalibrated or split or just don't fit, a lot of those, right, are sort of concentrated in those false positive calls, and especially, right, at particular places, right, where there are a lot of called m6A sites that are very close to each other. So when you're sort of at a crossroads and you don't know what to do next, you try and simplify the problem. And in this case, I looked at and tried to understand, right, why and how one particular site that had 100% wild type, 0% metal 3 knockout, like the perfect site, right? What about the perfect site did I need to learn? What could I learn from examining it that would help me investigate all of the sites within those box plots? And so if you look here, this is a specific K-mer, the specific position that's reported in F5C. The simplest question is like, okay, this position was reported as 100%, the bed file, but which adenosine in this sequence is actually m6A methylated? And it turns out if you look at the IGV and you look at the K-mer that's associated with the position, you can tell that the m6A methylated base is that first adenosine. That's what's basically being reported, and that's what matches the position. So the F5C position is 520138082. In IGV, it's 530138083. That's because IGV is a one coordinate base system. That means the first base in a genome is given a 1, whereas in the modbed or bed files, it's given a 0 position. So we can figure that one out now. Okay, so and then we can label it 5' to 3'. Great, and we get this F5C position basically represents one snapshot of the m6A methylated base in the pore. And then the question is, what snapshot? And this is the actual diagram. So I took this diagram, right? We know it goes in through the 3' end, is at the 5' end. And I made this exact, and we've already talked a little bit about this truncation phenomena. But I actually just basically took this, sort of put it onto the side and put it over the pore. And that gave me a sense, it's kind of a schematic way of doing it, but that gave me a sense of like where this A is, where this m6A is at this particular banded alignment for what is being measured, right, as an m6A. But it turns out, right, that the m6A position or this position is not the only place, right, within the pore that this m6A base is going to be measured, because ultimately that pore fits 9 bases. Originally they fit 5, but maybe they're affected by more longer range, right, interactions before and after. So there could be a lot of things, right? So if you look at, right, position -1 or position +1, you will see, right, that adenosine, that m6A adenosine and its location in the pore is gonna move. So in the position -1, you get that adenosine is going to be the second base from the end. And so for the subsequent analyses, right, if you want to understand and you want to look at all the signal as the m6A is in the pore, right, anything that might be associated with the m6A, you want to go to all the positions before and after. And so for the subsequent analysis, I pulled all the F5C k-mers in data from position -9 to position -2. And what you can then do, and that I did for every site that I measured, except for the ones that were below 80% for both, because that might have been computationally expensive. But what you can do is then take those k-mers and you can write from position -9 to position +2, you can take that signal that's reported and graph it out across these different pieces. Now, what's interesting is that, let's say, right, we did a position greater than 10 cutoff at the beginning of all of this, right, at least 10 reads, but that doesn't mean that the base 9 bases before had, right, the 10 read cutoff. So some of these, right, are low coverage, and it's hard to calculate statistics for those sites, 9 base pairs or so before the actual base pair of interest from F5C. And this could also be, right, because of the miscalibration reads, the other split read phenomena or whatnot. But certain information, so certain reads don't have data from position 9 to 1 for the whole range of what I'm looking for in the original signal to sequence alignment. So this is something that you look at and you basically say, exclude all of the things that aren't able to output this summary statistic data on the event currents between 9 positions before to 1 positions after. So the requirement, because you want to calculate a standard deviation that you're adding effectively, is that you need 2 calibrated reads. That means reads that have a signal to sequence alignment for each m6A site that you're going to investigate. And when you ask which sites are these that would be failing such a non-rigorous cutoff, it's really a lot of those false positive calls. So you're losing almost 50% while losing almost none of the true positive calls. Nevertheless, we still have, right, at the end of the day, a sort of hierarchy chart that looks like this. We're not calculating with the below 80%, but you've got these 488 sort of false positive hits, these 162 true positive hits, and this one lone warrior over there, knockout 80%, wild type less than 20%. So this here is just to give you a scale of some of the massive amount of data that these experiments generate, and the kind of SQL database processing and other optimizations that have needed to be done in order to accurately work with the data, handle it, and get meaningful reproducible outputs. But yeah, so SQL is another programming language that's typically used for database optimization that we've been trying to apply in our epitranscriptomic analysis. So back to these sites of interest. What differentiates a true positive from a false positive site? Now that we've moved non-calibrated sites, sites that had like funky coverage or whatever not, what differentiates these? And so if you look at the event standard deviation, what you see is that there's very little difference between true and false positive. That one knockout is going to have a lot of just variation because it's just one signal. But you can see on the average out, right, the true positive, false positive for event standard deviation has very little difference. For event length, there's also relatively little difference between this true positive cohort and false positive cohort. But what did emerge when you look at the event mean, what was really cool is that there's sort of this difference in the actual current. So the current, right, if you look at there's that dip in the green line of the wild type true positive, and that is sustained, right, as you get to the -4 position. So the -4 position is when that m6A is right in the middle of the current. When you get there, you see a dip, and it's sustained, right, from a particular mean level. You go to -5, you go to -6, -5, -4, -3, -2. There's this sort of beautiful dip in the current. So what this means, in essence, and yeah, if you look at the event mean, or if you look at it a different way, so this here is like the p-value difference, which just says that there's a change. And if you look at the actual mean there, you can see that it's higher in knockout. And ultimately, right, that in summary says that an m6A methylated adenosine generally causes a decrease in signal current when the m6A methylated adenine is in the middle of the pore. Ultimately, right, and this is something that you can say from first principles, having looked at the specific signal currents and the methylation currents and gone back to the original data and looked at all of this from a standpoint. Now, naturally, some might ask at this point, this was all a very roundabout story and way of understanding. Are you able to take this sort of knowledge, right, of m6A methylated adenosines cause a decrease in current signal when the m6A methylated adenosine is in the middle of the pore? Can you take that first principles knowledge and actually apply it and be able to tell, like, oh, this is a false positive hit and this is a true positive hit? And unfortunately, the answer is no. And that again, this is a general principle, but it doesn't apply to every specific k-mer that you sample. Might not apply to every specific k-mer, even if you had enough examples for all of them. It's only a general rule of thumb and not precise enough to differentiate totally true from false positives. So you could maybe generate more true positives, but you'd also be losing a lot of false negative, right? You'd be giving up a lot of data on the table and have a hard time measuring a lot of sites. The other question that may come to mind is if you go all the way back to this plot, right, that I had of knockout and wild type, and you compare that to like any of Nanopore's plots or any of Nanopore's claims, there's quite a large disparity in this, right? Why exactly would they, release a product that might look like this? And maybe, at the time that I did all of this, I was not aware of the answer, but now I am. And I learned soon after completing this investigation that Oxford Nanopore Technologies, as a part of, right, the modified base calling output, developed their own new metric for modified base calling. It's called the modification confidence score. It is output along as you can set basically when and how it outputs this score. but its output along with the modification models that you run for a particular sample, and it is a measure of base caller confidence in a modified prediction. It is an arbit, so I put this timeline here between 0 and 1, which looks very arbitrary, and in fact the prediction is very arbitrary. It's just a score between 0 and 1. It can be fractional, can be decimal, and you basically get this output. So okay, we have this modification confidence score that we learned about. Why did you get sort of all of this? It's because the modification confidence score that is utilized for this particular prediction, or that was used for this particular compilation in ModKit, which is the software that aggregates, takes the BAM files, aggregates per site the confidence in terms of the m6A site stoichiometry for that site. So when we ran the automatic version of ModKit, it estimates a base caller modification confidence score roughly, it depends on your sample, but roughly around 0.75, which in my experience is far too lenient and again allows the introduction of all of these false positives. But they basically have an internal algorithm that I consider lenient that estimates what base caller modification confidence you should use for your sample for when you aggregate the data. But you can actually set it yourself. And so having the metal-3 knockout and the wild type data, we were able to look at, right, the confidence. So if you look at regular adenosines and you ask the model, how confident are you in the calls that you made on these regular adenosines, looks the same between metal-3 knockout and wild type, which is good. Both models are looking at canonical adenosine. The model is looking at canonical adenosine in both samples the same way. That's important. And then when you look at m6A and the metal-3 knockout, you would expect there to be a lot less m6A versus in the wild type. But what you ultimately see, right, is that they actually have very similar distributions and very similar curves. And it's only when you get to that 0.99 or higher confidence, so much higher than the default confidence, that you start to see a difference in the model. So ultimately, right, it's a machine learning algorithm, so it's using a lot of multivariate signals and inputs in order to define, right, what is a true m6A from a false m6A. But we can use this benchmarking to help us better filter and identify what is true positive and false positive. And when you go from the 75% filter to the 99% filter, you can, not just sort of take these true positives, but when you look at now the plot of the wild type distribution versus the metal-3 knockout distribution, there's an actual very distinguishable difference, and that you get a lot more m6A stoichiometry that ultimately lines up with the mass spec data. And when you look at when you clean this up a bit and you look at just the Dratch motifs with the latest m6A all context model, so again from September 17th, the m6A Dratch, well, it's not the m6A Dratch model. I want to be precise. This is technically the m6A all context model, and then we only took adenosines that in the reference are Dratch motifs. It's rather, it's an incredibly tight correlation between the wild type and the metal-3 knockout. And you can see here, right, that the m6A stoichiometry level, right, matches what we see in Glory. So all of this is great, and it seems like we've arrived at a better place with the technical benchmarking. We have learned about this, what to me seemed like a mysterious and hidden modification confidence score statistic, unpacked its meaning, uncovered it, and we're able to apply it for this better. But there are a couple of challenges that still remain. I have not really listed them on the slide, but I'll try and describe them in general terms. So one thing is that M6A, as I mentioned, is 0.1 to 0.4% of adenosines, which sounds like a lot. But again, these are per molecule models. So even though, right, you have, let's say, right, even though you have, right, on a per molecule basis, if you go all the way back to the nanopore slides, right, a 96% accuracy on M6A. If you were to take, 10,000, right, M6A molecules, that still means, right, that 4% of them, right, 4% of those are going to be miscalled in some way. They're not called as M6A, called something different, right? There's going to be some sort of misnomer. Or on the flip side, right, for the adenosine, right, 98% of those are going to be accurate. But if you have 10,000 molecules, right, 2%, or I don't know if I want to do mental math right now, right? But um, I guess that would be 200. Yeah, 200 of them are going to get miscalled. 200 sites, 200 adenosines, 200 molecules. That might still be very meaningful in the context of there not being a lot of M6A or not being a lot of site level specific stoichiometry in the first place. So M6A is good because it has a lot of high stoichiometry sites, and we showed that we were able to capture a lot of those. But let's say you are interested in sites that have less than 10% stoichiometry. How are you going to be able to differentiate, right, between a true positive and a false positive call? At the end of the day, you have to find a way of being able to get per molecule. You have to get filters that are going to increase, right, your accuracy rate of your model and remove, right, false positives, control for false positives as much as possible. And this also is relevant for these other models, M5C, pseudouridine, inosine, because those modifications themselves are far rarer than M6A. So at the 99% level, what you get is a false positive rate of roughly 0.02%. And yet, the number of M5Cs, the true M5Cs in your whole transcriptome is less than 0.02% of all cytosines. So if your error rate, right, is for a per molecule basis is greater than the existence of that epitranscriptomic modification, how in the world are you gonna call it out and make meaningful data out of it? So we had a strategy for M6A, but at the end of the day, right, how do we push the boundaries? How do we make this workable and usable for pseudouridine, M5C, and inosine? And I guess I should have put in the slides the that show this phenomenon better. I might add these when I send these slides out because I think it's a very good demonstration. But we took two technical replicates, right? Basically where the same dish was split, RNA was extracted, and then these were compared. And if you look at M6A, right, a lot of the sites are shared, as you would expect, right, at the same stoichiometries. If you look at pseudouridine, if you look at inosine M5C especially, you get clusters, huge clusters of sites that are only called in one technical replicate, but not the other, which you shouldn't be seeing in this case, right? Because basically what if you are seeing like between technical replicates a lot of non reproducible results that suggests that, right, those are basically just the noise patterns that appeared in those particular samples based on that 0.02% error rate. So how do you ideally control for false positives in these nanopore direct RNA sequencing analyses? And what we were very lucky with was that about a month ago, somebody published a preprint. And then if you look on the PubMed version, but not the bioRxiv version for some reason, you can see the European Nucleotide Archive deposition for their original data. And they did an IVT control on these cell lines. In addition to sequencing, right, just some RNA 004 HEC293T samples or HeLa samples. What that means is that they took the RNA library, they converted it to cDNA, and then put it in an in vitro transcription reaction to convert that cDNA, all the cDNA, back into RNA. And that basically produces an unmodified version of the RNA library that you can use for all of this false positive analysis and discovery. And so what you can see, so for I'm just going to show the data for m6A here, is that the HEC293T IVT direct RNA, right, as you expect, as you increase the modification confidence, as you increase that sort of per molecule site cutoff, the number of modified nucleotides that get called decreases for both the HEC293T and the IVT, right? So you're removing maybe noise, false positive calls, lower confidence calls, whatever you want to call them, and you're sort of getting to more precise signals, more extraction. And and what you can see, right, for M6A, which is the most common modification with this with this sort of perfect IVT negative control, you're getting to basically like only 5.5 times enrichment, right? So there are only 5.5 as many M6A calls in your actual native RNA library that should have M6A over this unmodified IVT control. So if you're looking for stoichiometries less than 10%, right, you're going to get a lot of noise. This is, you could easily, right, depending on your coverage cutoff for a particular site, if you were looking at, for example, 20 reads as a coverage cutoff, you're gonna, only 2 reads off, and you've basically got a new site that's going to appear in your final readout. So again, coverage at each site is going to be, important to calling these things. But at the end of the day, right, the reason that the coverage, it's a function, if you increase the coverage stringency, right, you required each site that you wanted to call to have 1,000 reads. You're going to call very few sites. You're going to lose a ton of your data within the transcriptome. With 20 reads, you're already losing a lot of sites, right, that might have less coverage than 20 reads. So, basically calibrating the number of reads and the coverage for what you're going to use to call a stoichiometry is itself, right, like a numbers game and an important step. So all of this data that I'm showing you is based on a well, so this data actually didn't have a coverage statistic associated because we were just looking at this number of modified nucleotides, looking at these sites, and trying to ask, well, okay, this is sort of the data that we're getting. This is what we're being told with the per molecule modification confidence. How can we develop some kind of filter, site level filter that could work better? So ultimately, we did. And the curve looks something like this now. So if you noticed, right, with the original, so HEC293T native RNA library, we were calling approximately 20 million M6As and at this 0.9 sort of cutoff, and that went down to about 10 million. And with this new cutoff, right, we get to actually here 5 million with so 4 times less at the 0.9, but this is a dramatic reduction in the IVT in what's being reduced with this filter. And you can see when you graph the ratio, so by the time you get to, you're just under 4 million by the time you get to the 0.99, but you've got 8,000-fold enrichment of the number of calls in your native library to your IVT library. And that is sort of to the point where you can actually start to look at lower stoichiometry sites and variances and assign them any particular meaning. And so for a lot of, your single molecule projects, this is the filter that I'm gonna have you implement and use in order to make your comparisons and to understand and underlie this data. And if you ask, like, what's, I guess, the secret sauce? Like, what is the game changer that causes this dramatic difference in being able to measure M6A stoichiometry? It has to do with looking at the individual distributions of calls within a particular site or k-mer motif. So what is known, of course, is that for certain k-mers, for certain sites, we looked at those, we looked at those squiggles, we looked at those lines. For some sites, it's just easier to measure whether the m6A methylation is present or not, and easier to differentiate that based on the modification confidence scores. And that ultimately has to do with the the way that the training set data set was constructed by Oxford Nanopore Technologies, factors that we don't know the inside details about, but that we can sort of start to back calculate from the data and the models as we're given them. So if you look at every site and you say, okay, I now want to make a curve at that site for all of the modification site level stoichiometries, whether they passed or failed my filter, well, you just look at the site and you say, okay, here's the sort of histogram plot. I have 50 calls at like 0.75 confidence and 3 that are above 0.99. When you look at that site, well, you see that, some of them along this distribution, most of them are really low confidence. Most of them, right, if you look back all the way here, right, most of them are like backed up here in these regions, right? And there's maybe a couple calls that end up passing, but, most of the modification calls at this site were low confidence calls, and we should eliminate them. And so the site level filter says for every site, is the number of m6A calls, is the number of modification calls that are greater than 99% that pass my per molecule filter, is that greater than the number that failed the filter plus 1? So at the end of the day, what you're asking for is, okay, we get all the m6A calls, we get the per molecule m6A calls that are the model is confident about, but then we only take m6A at sites that are enriched for molecules with confident m6A methylation calls and confident canonical adenosine calls, and then comparing those across the cohorts and the distributions. And after lots of technical benchmarking, looking for samples, generating samples, sequencing, combining, we have a sort of strategy and a filter that will allow us to sort of broaden our horizons and be able to do, right, what is sort of the promise of this, which is being able to call, going back to sort of the middle or the beginning, depending on how you look at it, of high throughput epitranscriptomic calling. Being able to take a sample, sequence it, get M6A, and trust it, believe it, and be able to use it for all of your applications, whether it's low stoichiometry M6A or otherwise. As long as, right, you have enough reads at your particular site. So I will also note that this project and some of its associated things has received, we've received lots of grants, lots of collaborations, lots of data. And this access grant in particular, for those of you who are looking to do single molecule sequencing and would like HPC resources, will be available. I can basically add you to the plan if you make an account on NSF Access, and I can add you to the Purdue Anvil CPU and GPU resources to help with computational processing. And that depending on like so ultimately all of the base calling should be done for those projects will be done in advance for you, but the CPU time might still be useful for working with the modbed files and the like.
Yeah, very GPU and CPU intensive, yeah. So if people you might not have pain in the class for the homework, but if you do a lot of it for lab work, you'll need it, yeah.
And yeah, so the only reason I'm going to do all the base calling for any samples that you work with pre-base calling is, and not like have you do it on the Purdue Anvil GPU, is because the GPU resources that they offer also have a 3 to 4 week wait time, which would not exactly fit in with the timeline for this class. So, but the CPU resources are very available. They should be readily available, and are much less used than the GPU resources. And I know you're taking the class, but I might as well advertise the class to you. I put this in every presentation. So yeah, you're taking the class, and I guess take the class. And here are all my acknowledgements. So thank you very much, and I look forward to your questions.